It gives me a great pleasure to welcome Dr. Andrew Balber from Takeda Biopharmaceutical Consulting, who will explain the principles behind ALDEFLUOR-based detection of ALDH bright cells, and specifically in the field of regenerative medicine. Prior to establishing Takeda in 2010, Dr. Balber was the founder and for 10 years served as the Chief Scientific Officer of ALDH Incorporated, a company developing cell therapies for use in cord blood transplantation and for cardiovascular indications. Dr. Balber helped develop ALDEFLUOR, a research-use-only product for identifying and isolating ALDH bright cells, and obtained FDA clearance to market a second product, ALDECOUNT, for clinical enumeration of these cells. Most significantly, Dr. Balber helped ALDH establish and maintain a clinical program during which patients were treated under 7 cleared INDs with products composed of ALDH bright cells for indications related to cord blood transplantation, critical limb ischemia, ischemic heart failure, and stroke. And let me now hand you over to Dr. Andrew Balber.
Thank you, Andrea. And thanks, everyone, for tuning into this webinar. I'd like to start by basically outlining what ALDH bright cells are. These are populations of cells with high levels of an enzyme, Aldehyde Dehydrogenase. You see a picture of some of those cells on the right there. You notice that there's a cytoplasmic fluorescence. That, in fact, is where the presence of the enzyme can be detected and what the products we'll talk about. Cells can be isolated from normal tissues by flow sorting with the ALDEFLUOR product that Stem Cell Technologies makes available. And they are highly enriched in cells expressing stem and progenitor cell surface antigens and functional activities. These cells meet all the criteria for stem and progenitor cells in a variety of different contexts. They can proliferate at the same level of differentiation, and they can differentiate into other kinds of more dedicated cells, tissue cells. In addition to this progenitor cell activity, they also have paracrine activities. They secrete cytokines and other factors which make them useful in regenerative medicine. And these factors, these activities, both the proliferation stem cell activity and the paracrine activity of these cells, have already been demonstrated in clinical trials to be useful in regenerative medicine. These early trials have proven the safety and the ability to make these cells in clinical grade and use them in the clinic. We'll get to that at the end of this webinar. I'll focus on regenerative medicine today. Here's what we'll talk about. First, I'll explain what aldehyde dehydrogenase is. Then I'll explain how ALDEFLUOR works to isolate these cells. We'll review briefly what kind of progenitor cells have been enriched using ALDEFLUOR, and then go through their potential in regenerative medicine. So turning immediately to the enzyme, aldehyde dehydrogenase, what is it? This is the general way that a biochemist would answer that question. We've got the enzyme, ALDH, which catalyzes the pure dinucleotide-linked oxidation of an aldehyde function to a carboxylic acid. And we've shown the carboxylic acid here dissociated at neutral pH, and that's going to be an important issue later on. So in a test tube, this is what ALDH looks like. The story gets a little more complicated if you start to talk about ALDH in particular, because there is no one ALDH. It's in fact a superfamily of closely related isozymes. These proteins are very ancient. They've duplicated through evolution, and as they've duplicated, they've taken on different functional attributes, and it's very important to realize that these functional attributes have now diversified in very many different directions. So we'll talk a lot today about what the relationship is between ALDH and physiology in stem cells, and that'll be a thing we'll have to explore more thoroughly. So different species have very different numbers of genes. That's what's happened in evolution. For example, humans have 19 functional isozymes for ALDH. Rats and mice have 20. Zebrafish have 15, and so on. And in addition to the functional genes, there are a number of pseudogenes, and there's some splice variants that come out in different tissues. So in using ALDEFLUOR in stem cell biology in different species, it's important to realize that different enzymes may be involved, and these enzymes may have different functions. All of these enzymes have a very similar catalytic site. They have an active site thiol that's been involved in binding the substrate, and a cofactor site for the pure denucleotides. The reaction mechanism is fairly highly conserved throughout this group, but what's different are the way that the substrates get access to this active site. There's now good evidence, and you can look at this reference that is on the slide, very recent evidence, discussion of this, is that there's a substrate channel that at the first level size excludes certain kinds of large bulky substrates in some ALDHs, and in other ALDHs allows these substrates access to the catalytic site. It's a very crude kind of way of putting it because it's not only site exclusion, but there also are amino acids along that channel that align the substrate so they can interact appropriately with the conserved active site. But nevertheless, this means sort of divides the ALDH family into large and small substrate channel and this determines what substrates can get in there. And since the substrates are what generates the physiological activity of this enzyme, that determines the physiological function of the enzyme. In the context of ALDEFLUOR, it's also important to remember that that substrate channel also controls access of inhibitors to the active site. And an important part of the ALDEFLUOR reaction is being able to inhibit it so that you can tell the difference between enzymatic changes that you measure by flow cytometry and non-enzymatic changes. I'll go through this in a minute. So the ALDEFLUOR product has been developed from mammalian cells. It uses an inhibitor that works on mammalian cells. It works on some enzymes in mammalian cells, but not all of them. In other species, the situation is quite different, and for that reason, you need to know a little about the physiology, and you need to use the product carefully when you're outside of the mammalian context. The full range, even in mammals, of which ALDH isozymes are responsible for oxidizing the ALDEFLUOR substrate, which is the substrate for ALDH in the kit, is not known. So no one has looked completely through all the 19 ALDH isozymes in humans, for example, to see which ones of those are oxidized by ALDEFLUOR. So, I mean by ALDH, which ALDH enzymes oxidize ALDEFLUOR. We'll talk about this as we go through and discuss the ALDEFLUOR product in a little more detail, try to hone in on that. So what substrates do ALDH oxidize in stem cells? What's the connection between a high level of ALDH activity and stem cell function? Well, there are two primary hypotheses in the literature. One is that ALDH controls gene expression indirectly by synthesizing retinoic acids. There are, in fact, 4 human ALDH isozymes, ALDH 1A1, 1A1, 2A1, 3A1, and 8A1 that oxidize retinaldehyde. Retinaldehyde is a bulky substrate, and in the reference that I cited on the last slide, the argument's made that the ability to oxidize retinaldehyde by having an open substrate channel is a very important change in metazoan evolution. It allowed these enzymes to use retinaldehyde to make retinoic acids and allowed retinoic acids to become a major morphogen in developmental biology. So far, when people have looked at ALDH bright cells made by using ALDEFLUOR, they have found, when they've looked at gene expression data, that each cell population expresses one, and so far only one, of the ALDH 1A subfamily gene products. That is, they've looked at, in several papers now, looked at all of the 4 retinaldehyde oxidizing ALDHs and only found one of them expressed, and it has not been 8A1. So far it's been 1A1. That's been found in human umbilical cord cells. 1A1's also been found expressed in human bone marrow, ALDH bright cells. And 3A1 has been found now in human breast cancer tumors that have a very high aggressiveness. In other species, 1A1 has also been found to be expressed, for example, in mouse pancreatic ALDH bright cells. 1A1 is the product expressed. So there seems to be something about being able to oxidize retinaldehyde that's important in these cells, and that only one of them so far has been detected. Retinoids work by binding to transcription factors, retinoid receptors, and altering their affinity for promoters on retinoid. There are certain elements on promoters called retinoid responsive elements that bind these transcription factors. And by altering the affinity of these transcription factors for promoters, retinoids can turn gene families on, turn them off. So internally, in a single cell type, the physiology of a single cell can be changed in that manner. Retinoids are also powerful between cells. Retinoids can be secreted, and they're powerful morphogens in developmental biology. For example, there's a field of retinoid concentration that's developed in the retina, in the developing eye, and that determines the morphogenesis of the eye. And in stem cells, it's well known that retinoids regulate stem cell proliferation, development, and function, and they do that in different ways in different stages. For example, retinoid depletion can stop the development of very early blood stem cells, but later on, retinoids are actually a developmental factor that promote differentiation. For example, in certain leukemias, retinoids are used actually to cause those leukemic cells to differentiate into cells that don't divide anymore. Now, that's a major focus of interest in ALDH and what it's doing in stem cells. But ALDH molecules do a variety of other things. The other major thing that's been talked about in terms of what they may do in stem cells is protecting these cells against oxidative stress. And in fact, it was this discovery that ALDH protected blood stem cells from the effects of cyclophosphamide that actually led to the discovery that ALDH was expressed in a variety of different stem cell types. So oxidative stress, for example, from oxidized lipids, from xenobiologicals of a variety of kind, from being in environments where the cells are exposed to high degrees of oxygen. For example, in the eye, in the corneal epithelium, there are many ALDHs expressed, and these help protect the eye from oxidative damage. It's important to realize this, because this means that not all cells that express ALDH in high levels are stem cells. There are physiological niches where cells use ALDH for other functions. And you have to, again, using this method to detect ALDH bright cells, you have to be careful to understand the physiology of the cell population that you're looking at. And in fact, when people have looked at limbic cells from the cornea and sorted them on the basis of ALDH bright cells, in that context, the ALDH cells that express most ALDH are in fact not stem cells, but they're in fact differentiated corneal epithelial cells. Another point to remember here is that some of the same ALDH isozymes that oxidize retinoids also oxidize other small toxic aldehydes. Several members in the ALDH1A1 family, A3A1 for example, can oxidize cyclophosphamide and can oxidize some small aldehydes also. So one ALDH isozyme might have different functions that can be important in stem cell biology. Also important here is to note that ALDH molecules are often co-expressed in stem cells at any rate with ABC, ATP binding cassette, transport, or efflux pumps. These are the molecules that have been first isolated as multi-drug resistance pumps, but physiologically they can pump a variety of toxins out of cells. And there's been some speculation that the co-expression of ALDH and these efflux pumps have to do with the ability of, or the need of stem cells to have some toxic molecules, including toxic oxygen products, pumped from cells, toxic lipids, and so on. These are all speculations at this point, but they're things to keep in mind when considering what ALDH may be doing in stem cells. Finally, ALDH molecules have other activities besides these two. They're involved in biosynthesis. For example, ALDH molecules are involved in folate synthesis, and folates, of course, can control the rate of proliferation in cells, which is important in stem cell biology. They also bind hormones. So there are other activities, and one should keep an open mind about what ALDH, at high levels of ALDH, might be doing in cells. So then what ALDH molecules are detected, we don't know for sure, but how does ALDEFLUOR detect them and help the isolation of ALDH bright cells? Here's the product. It's a reagent system that allows the enumeration, detection, and prospective enrichment of adult stem cells from normal tissues. That's what we're focusing on today. And the reagent system includes a substrate for ALDH and other ancillary reactions that reagents that we'll go through in a minute. It detects viable stem cells, and I'll tell you why that means. It's an important factor of the cells. You don't tend to pick up, don't pick up late necrotic or apoptotic, late apoptotic or necrotic cells with this method. The reagent system does not impair the functionality of these cells. In fact, it enhances stem cell activity when you purify these cells away from other cells in the product. And it's not transferred. The reagent system is not transferred in large amounts that impair stem cell function into an animal or a culture dish or in the clinic into a patient in high concentrations, which is an advantage, for example, over some antibodies which may remain bound to a cell, for example. So to look at how the product works, we have to take the ALDH reaction that I showed you previously in a test tube, and we have to put it in a cell so it becomes a flow method to detect the enzyme in a living cell. And this cartoon does it. In this cartoon, this box is a cell membrane. Here's the substrate for the aldehyde dehydrogenase reaction. First step is it has to be able to get into the cell. Second step is that it gets oxidized, as we saw before, with pyridonucleotides. But this means in the cell that it's going to be sensitive to the oxidation state of the cell. There has to be NAD around to oxidize it. It's going to be dissociated into the carboxylic acid. And normally in a lot of cells, this carboxylic acid, because it's charged, will be retained. As I've said before, in some stem cells, there are these ATP binding cassette proteins. These proteins can actually cause the efflux of the reaction product. That's an energy-dependent step. So in the ALDEFLUOR product, when using it, you have to take steps to block this activity in order to retain this product inside the cell. I should mention here that I've drawn this as if it's a cytoplasmic cytosolic enzyme. And many of the ALDHs that are important in stem cell biology, certainly the ALDH1A1 family that I talked about, is a cytosolic enzyme. The picture that I showed on the first slide showed a cytosolic fluorescence. Whether or not ALDHs in other organelles can in fact oxidize the ALDEFLUOR substrate is not known. But so far, everyone who's looked at a microscope has described the fluorescence as cytosolic, and we'll go with that and say that this cartoon actually represents what's going on in this cell. So here's the reagent. This is the ALDEFLUOR reagent, which is the substrate for ALDH. Let's start down here with the real substrate. Here's the aldehyde function. This is BAA, which is BODIPI, which is the green fluor that's attached to the molecule. Amino acid aldehyde. Here's the amino group acetaldehyde. This is the aldehyde that gets oxidized. This is the fluor. This is a linker. It gets oxidized to the carboxylic acid. I've shown it protonated here. This is the reaction product. In the kit, in order to stabilize this reactive aldehyde so it's stable for storage, it's a dry down product stored in this form with a stable leaving group on here. To use the product, this is dissolved in DMSO. This is a very hydrophobic molecule that needs this organic solvent. It's acidified. You get an elimination reaction on this carbon, and you end up with the aldehyde function. So this is used just before you use the product. This is activated in the tube, and off you go. Notice that this is a pretty bulky substrate. It's got these halogens sticking on it. It's got this big ring. It's very different from, for example, acetaldehyde. And remember what I said about the substrate channel, taking on bulky substrates and doing some size sieving among the different ALDHs. So we're trying to talk about how this works. Here's how it works in the actual tube. Here's the activated BAA reagent. It can go into the pool and into the cell because, as I said, it's a hydrophobic molecule and it forms a pool. That will be green because of the substrate that accumulates. If you use this in the presence of an inhibitor that stops the reaction to form the product, then you will only have fluorescence from the pool molecule. In another tube, the pool now can be oxidized by LDH to make the ALDEFLUOR product, which accumulates in the cells, and the cells will turn green in the cytoplasm. Now, I said that there were these ABC transporters that could efflux this. In the ALDEFLUOR buffer that one can buy with the product, there are inhibitors that block that efflux in stem cells. Now, in some cells, of course, the ABC transporters that are in other species may not be the same as they are in mammals. In that situation, other steps may have to be taken to block efflux. You can do that simply by putting the cells on cold or putting in an energy inhibitor like ALDEHYDE, and that would keep those cells green, so because this efflux doesn't happen. There's one other possibility here for how the cells can lose the product, and that is that they can die. If the cells become late apoptotic or necrotic, of course, the membranes would be open and that reaction product would leak out. And this is the reason why you don't see in the ALDH bright cells that one scores as highly fluorescent with the green fluorescent. There will not be late necrotic or apoptotic cells because these cells lose their product. So what does this look like actually in a what does this actually look like in a flow cytometric assay? One does it in several steps. First, one sets up a cytogram with forward scatter on the one axis and side scatter and gates on nucleated cells. Then one, on this gated area, one takes side scatter versus green fluorescence in the tube that has DEAB, the enzyme inhibitor, and sets up a region for cells that are low in side scatter over here and high in fluorescence, green fluorescence for the BODIPI over here, and compares that to the tube without DEAB. And here are the cells that are defined now as ALDH bright side scatter low. These are our ALDH bright cells. In principle, what's happened then is that these cells have moved from this population here out here because of the enzymatic activity. Notice it's a big increase in activity in fluorescence. It's 2 logs increase here. To show you that these cells here are in fact this fluorescence that's in these cells is in fact due to the substrate pool I mentioned a minute ago. You can look at this tube with no substrate at the same photomultiplier settings, and you can see these cells are all gathered back at the axis, whereas this is all, these are all the cells without any substrate in the pool. So these are the cells that we're interested in. And what we can do now is we can sort these cells. We can use them first just to identify those cells. We can count them if that's all we're interested in doing, say how many are in a preparation. But we can also sort them to give ALDH bright cells, which we can subsequently use for physiological studies on stem cell biology. We can put them into culture and ask if they have the attributes of stem and progenitor cells. For example, we can do colony-forming assays. You can do various kinds of sphere-forming assays, such as neurosphere assays, mammosphere assays, and other kinds of sphere assays. You can actually go right into animals and ask in animals, can they form tissue in appropriate animal models? Or you can do an expansion, for example, in vitro under conditions where you expand progenitors and then drive them with cytokines and then put them into an animal model or back and forth. So this is the basic protocol, and I should add here that as well as animal models, not with ALDEFLUOR because it's an RUO protein, but with the same technology, one can substitute a person here. Now, in principle, this shows that you can get fairly good isolation of ALDH bright cells simply by sorting using ALDEFLUOR. Whether or not you want to do this depends on how long you want to sort, what kind of purity you need for the experiments you're going to do, and basically how you want to organize your work here. One thing one can do with ALDEFLUOR, of course, is to put other kinds of debulking steps in here, for example, depletion with immunomagnetic beads of some kind to reduce the sorting time. There's a lot of flexibility here. The other thing one can do is to put in other antibodies here. You can use multiparameter sorting here to detect subpopulations of these cells. For example, you can do that up front, or you can do that subsequently. You can identify, for example, ALDH bright CD34 positive cells in this population, and ALDH dim or ALDH low CD34 populations in this cells in this population. And in general, if you read the literature on ALDEFLUOR, you'll find that people do define different cell populations. They can call side scatter low ALDH bright dim, sites scatter low, ALDH bright, and so on. Some people will actually define all the other cells that are not ALDH bright as a dim population, and I'll use that kind of language as I go through the talk. So what kind of progenitors do you get that way? The first important point, and I'm sorry this is in the way, let me move it down here, is that ALDH bright cells are usually adult lineage-committed progenitors. There has not been a lot of work done to nail this down, but I think most of the evidence that there is consistent with this. There have been some studies showing now that embryonic stem cells are usually ALDH dim prior to being differentiated. If one looks at ALDH bright cancer cells, they usually retain expression of markers that are characteristic of parent tissues, for example. This says that it isn't a very early sample, a very early undifferentiated cell. There's no evidence now for tumorigenesis in animal models, for example, teratomas, in animal models of tissue repair using ALDH bright cells that have been isolated from non-cancerous tissue. Right? These cells differentiate appropriately when they get into tissue environments different tissue environments when they're transplanted into animals. One doesn't see tumors running up. This is consistent, in fact, with the physiology that's been done in mice and zebrafish, showing that knockdown of ALDH expression either genetically or with RNA silencing or inhibition of ALDH expression, for example, in zebrafish is routinely done with DEAB to make phenocopies. Knockdowns tend to stop at particular stages of embryonic development that are past blastomere stage to develop tissues. For example, in mice, knocking out specific ALDH isozymes can stop things stop development at the formation of the neural tube, not very early on. The only example that I know of that's different from this is in the tunicates, which is very early, which is about the time that some of these retinoid pathways have started to develop and diversify. And in tunicates, it's actually quite interesting in that ALDH bright cells are totipotent, and then a whole embryo can be formed from them. But in general, in most of the other animal models that we've studied, these are lineage-specific adult progenitors. So what kind of progenitors are they? Most of the information that's available has been obtained for a long time now from hematopoietic, and certainly from mouse, from human, from zebrafish. If you sort ALDH bright cells from a variety of tissues, in humans it's been done from cord blood, mobilized peripheral blood, and bone marrow, you get an assortment of hematopoietic stem and progenitor cells at different stages of differentiation that form colonies. The most recent, maybe most comprehensive paper on this comes from Dr. Storm's lab very recently, where they've shown for the first time, in fact, that the ALDH bright CD34 population can in fact give rise to T cells as well as all the other lineages in blood, which completes the picture. So all megakaryocytes, both kinds of lymphocytes, NK cells, granulocytes, monocytes, erythrocytes, they all can be formed from ALDH bright cells. The balance of those different progenitors with very early stem cells depends on what kind of tissues one gets. Similarly, mesenchymal progenitors have been isolated from ALDH bright cells from marrow, from adipose tissue, and from cord blood from humans. These are multipotential mesenchymal progenitors that can give rise to all the cell types that mesenchymal cells can give rise to. Endothelial progenitors have been isolated from human marrow by two groups. Neural progenitors have been isolated from mouse and have been detected in rat using ALDEFLUOR. In the mouse studies, they've been detected in marrow and in fetal brain. In the rat, they've been observed in the developing spinal cord at the time that the spinal cord, the neural progenitors are moving into the spinal cord and the cord is closing. Mammary tissues. The ALDH bright cells from mammary tissues and biopsies, from cell lines and biopsies also are normal, give rise to normal ductal mammary tissue and endocrine tissue. Skeletal muscle progenitors in human muscle biopsies. Mouse pancreatic duct tissue, multi-potential progenitors for pancreas formation from the mouse. Prostate epithelium from the mouse, and also kidney proximal tubules from humans. I think this is a fairly comprehensive list, and stem cell will will provide you with references for these things. So it's a broad marker. It's remarkable. It's remarkable in the different kinds of lineages that share the use of ALDH that can be detected by ALDEFLUOR and isolated by ALDEFLUOR. And one of the general characteristics. Well, I've already hinted at some of these things. Populations isolated from different tissues are different in composition and function. That they are different stem cell antigens. They're highly enriched for appropriate canonical stem cell surface markers characteristic of the lineage in that tissue. CD456 in muscle cells, CD133 in the tubules, CD34, 133 in blood, and so on. They are heterogeneous, as I've said. When people have looked, they have found that early transcription factors, such as octamer, SOX, a few of the other early transcription factors that have been associated with stem cells are expressed in these cells. And they find always that the ALDH bright cells that have similar surface antigens, these canonical surface antigens, have higher functional attributes of stem cells than ALDH dim cells that express the same surface markers. For example, ALDH bright CD34 positive cells are multilineage skid-nod reconstituting cells. ALDH dim CD34 positive cells only have short-term reconstituting activity in the skid-nod mouse model. So the same has been found in muscle cells, that you've always enhanced and enriched the stem cell activity by some proportion in the ALDH bright fraction. So ALDH bright as a marker does say something about the stem cell activity in a stem cell population, and it says more than the surface marker does. Now, all those studies that I've just reviewed have to do with progenitor cell activity. They've looked for colony forming, sphere formation, formation of tissue when transplanted into animals, those kinds of standard, can you make a tissue kinds of models. This is a different question. Can you repair a tissue, a damaged tissue, using those cells? And I'm going to talk about a few examples that show that ALDH bright cells can in fact repair damaged tissue, and this is where it gets interesting in going to the clinic. In the context of neurological diseases, Dr. Cordian's group in Milan has published a series of papers showing that ALDH bright cells from mice, they can either be isolated from fetal brain or from bone marrow, and either used directly or cultured. And a lot of these studies were done with cultured cells that were expanded in culture and then driven down appropriate pathways with cytokines, can regenerate spinal motor neurons that innervate the hind legs of mice and rats. These cell populations are multipotential neuroprogenitors, and they can, when gone intrathecally, they can migrate down to an appropriate region and help form the formation of these motor neurons that actually grow out from the spinal cord to the appropriate position on the hind limbs. It's a remarkable set of studies. They did show that these cells proliferated and could actually contribute to the neurogenesis in these animals, but as I'll mention again in a minute, they also showed that these cells could have paracrine effects that could induce endogenous neurogenesis in these animals. Similarly, human cord blood cells, when injected into non-skin animals, can help protect these animals from death following acute intoxication, where the animals were given high doses of carbon tetrachloride. They were protected from death, and ALDH cells human cells derived from the ALDH bright cells were found to infiltrate the liver, although they did not participate necessarily in forming new liver tissue, again suggesting that there was some paracrine effect that they were helping in this liver regeneration model. A very recent paper that was just been posted on the web from Dr. Hess's group in Toronto has shown that ALDH bright cells from human bone marrow in the not-skid model can help in the restoration of pancreatic islets following streptozotocin treatment. This was a very interesting study in that the ALDH bright cells actually contributed to the regeneration of islet function primarily by angiogenic mechanisms. They helped to restore blood vessels and vascularization and help these the islets that were left grow. And that way they enhance the effects of mesenchymal cells, which couldn't do that. Mesenchymal cells had a different effect on pancreatic islet formation. And there was a synergy between the two cell populations in this model. The important part from what I'm going to say in a minute here is that there's some evidence here that ALDH bright cells from bone marrow are in fact angiogenic in this context. And this gets to the following three models, all of which have shown that human ALDH bright cells are angiogenic in appropriate disease contexts or injury contexts. In each case here, an acute ischemic injury was induced in a mouse, in a skid-knot mouse in fact, either by ligation of the descending cardiac artery, ligation of the femoral artery, or occlusion of the middle cerebral artery. These acute ischemic events were then followed by infusion of ALDH bright cells in the heart intravenously, in the femoral artery model, high-level ischemia model, and also intravenously in the stroke model. It was through the carotid artery. In all cases, labeled cells were found to go home very specifically to the site of ischemia, to stay there for a couple of weeks usually, a week to two, and then to induce angiogenesis at the site of the injury. The induction of angiogenesis in some of these models actually caused an increase in perfusion at the site of injury, which of course would mitigate the damage that these tissues were undergoing. What's interesting in all of these situations is there didn't appear to be human cells actually forming the vessels, the new blood vessels that were formed, or any of the other tissues, but they did seem to stay near the vessels but not actually become part of them. So it looks like they were operating by paracrine effects. So in all of these models then you've got evidence for paracrine effects, although in the mouse model you've got some direct evidence for actually proliferation of the cells contributing to new tissue. But there are two possible ways. Certainly in anything having to do with blood transportation and hematopoiesis, the ALDH bright cells are actually proliferating and becoming the blood cells. Different models. Let's dig in to this high-level ischemia model a little further, because I think it's very interesting and it says what ALDEFLUOR may be doing and why it's useful both for research and in the clinic. In the high-level ischemia model, the evidence in the reference that's given here is very clear that basically the entire angiogenic component of human bone marrow is captured in the ALDH bright cells. Dose-wise, 100,000 bone marrow cells had a strong angiogenic activity, but 500,000 dim cells from the same marrows had no activity. It's also interesting to make the point I made before about ALDH being a marker that's different than CD34 or CD133 or 105, or these canonical stem cell markers. About 75% of the CD34 positive, or 133 positive, or 105 positive cells are in the ALDH dim fraction in these experiments, and nevertheless they had no activity. So again, it's the ALDH bright varieties and flavors of these markers, cells expressing these markers, that are important. And finally, this is a critical point. The ALDH bright cells that are enriched through ALDEFLUOR sorting had more activity than unsorted bone marrow. And even though the unsorted bone marrow contained 4 times more ALDH bright cells, so the other cells in the unsorted bone marrow can inhibit the effects of ALDH bright cells in this angiogenic model. This is critical, and this actually argues why one would want to use sorted cells to study the potential effects of these cells, or put sorted cells to, in an animal model or in the clinic to control the physiological activities that these cells have in the absence of other possible antagonistic effects from other cell types. So what could some of the paracrine effects of ALDH bright cells be? And we'll focus here on some of the work that the Aldehyde group has done looking at angiogenesis as an example. Here's one set of pictures that make it. In this picture, ALDH bright cells have been labeled with a cytoplasmic label. They're green. It's not with ALDEFLUOR. And what they've been done is mixed in a culture with UVAC cells. These are the UVAC cells that form these networks on basement membranes. And if you look at an overlay under normoxic conditions, you see that the ALDH bright cells are sort of randomly distributed. But under hypoxic conditions, which mimic what will be going on in an ischemic injury, the ALDH bright cells are closely associated with the network and can adhere to hypoxic endothelium. This association with them could be signaling. It could be a signaling interaction, but the ALDH bright cells can also work with hypoxic cells in a paracrine kind of way. Let's focus on this channel. Here's a HbAC network of endothelial cells. Here's what happens to that network if you make the cells hypoxic overnight. These branch points break up, cells die, the network shrinks. But if you put either a conditioned medium from ALDH bright cells from bone marrow into this culture, or if you put ALDH bright cells across a transwell from these cells, that's shown by the arrow here, these networks are maintained under hypoxia. So it is a protective environment. There is a paracrine kind of effect that ALDH cells can mediate to stabilize endothelial cells. Numerically, this is what that data looks like. Under normoxia, the ALDH bright co-treated, ALDH bright co-cultured and HbAC alone are about the same in terms of network branching. But in the situation of hypoxia, there's a huge statistically significant difference between the ratio, the amount of network branching that goes on in hypoxia without ALDH bright cells and in the cold cultures with ALDH bright cells. So they do protect ischemically damaged or hypoxic damaged endothelium. If you look now at the cell populations, the ALDH bright cells that have been cold cultured with normoxic or hypoxic HbAC cells, this is a gene expression array. You see that there are a number of genes here that are expressed at the same level in both cultures. But this collection of red dots shows that there are a list of factors that are angiogenic factors, factors that control the formation of new blood vessels. They include cytokines, receptors for cytokines. They include signaling molecules, for example, ephrins and ephrin receptors. They include VEGF, the cytokine. They include adherence molecules and the whole gamut of families that contribute to angiogenesis. These are now being upregulated in the presence of hypoxic endothelium. And the aldehyde group has also shown that the presence of the ALDH bright cells also changes the expression of angiogenic factors in the endothelial cells. So these cells can work by paracrine mechanisms as well as being endothelial cells. To summarize then, what can happen here is if you get bone marrow cells that have ALDH bright cells in them, you sort them with ALDEFLUOR. These cells can now go into a system. They can bind to ischemic endothelium. They're upregulated, they're angiogenic molecules. They can be progenitors, and all this is focused at the site of an ischemic damage because the cells can home to that site of damage. The other cells in bone marrow, for example, can compete by either competing for homing or by making antagonistic cytokines. That would inhibit the formation of new blood vessels in this ischemic area and therefore inhibit reperfusion of the ischemic tissue. So, will that work in the clinic? One sort of piece of evidence that maybe when we're dealing with cells that could be clinically important comes from clinical research which is correlating the effects of different cell preparations, cell products, with the number of ALDH bright cells in them. There's now a good deal of activity looking at ALDH bright cells, the number of ALDH bright cells in cord blood units as a measure of cord blood potency. This is going to a clinical trial fairly soon. This is similar to what's already been done to measure the efficiency of mobilization through counting ALDH bright cells that come out in a mobilization product during the apheresis project. This has been done, for example, by the group at the University of Arkansas for a while now as a way to measure mobilization in their patients. Similarly, there's been literature from Germany where Dr. Fesse and his group have looked at the ALDH bright cell counts have been used to measure the integrity of frozen bone marrow products when they're going to need to be used for a second transplant. And again, the dose of ALDH bright cells that are in these products are good measures of good predictive measures of how fast these patients will re-engraft. It isn't limited, however, to hematopoiesis. A group at Duke, headed by Dr. Pavsik, has published couple of papers now showing that the number of circulating ALDH bright cells in peripheral blood is inversely associated with the risk of a recurrent heart attack in patients with cardiac disease. Basically, the notion here is that these ALDH bright cells that can circulate in peripheral blood, many of them are in fact circulating endothelial progenitors, and this group has shown that, and that the more progenitors that are circulating in a patient, the better chance they have of not having another heart attack. This is number of ALDH bright cells has actually been used now by that group to monitor clinical trials with agents that are supposed to improve cardiac health. Finally, the Duke group has also shown in the clinic that in elderly patients, ALDH bright cells exist in bone marrow in healthy elderly patients who are coming in for hip replacement surgery, even though they may have fewer circulating stem cells, they still have a good reservoir of ALDH bright cells in their bone marrow. Suggests that maybe looking at bone marrow using ALDH bright cells would be a good way to identify patients who are good or maybe poor candidates for cell therapies that depend on a source of good stem cells in the bone marrow. Now, these are all clinical research. Some of these are being used. In some cases, people are looking to use ALDH bright counts as good clinical assays to make decisions on patient care. And in that context, should be aware that there is a product that embodies the ALDEFLUOR technology, but in a regulatory wrapper that in the United States is cleared by the FDA for in vitro diagnostic uses with fresh and frozen peripheral blood, cord blood, and for bone marrow. This is ALDECOUNT, which is another product that's available from Stem Cell Technologies. The technology is the same as ALDEFLUOR, but beside the regulatory wrap, it's formatted differently for clinical use. You can see in this case that there are two boxes, and basically this is a box of tubes that are set up so you can have one tube use as opposed to ALDEFLUOR, which is a bulk tube use. It can be used prospectively in clinical practice, retrospectively in clinical research, as I've just discussed, and it's not suitable for cell sorting because it uses Aldehyde as the efflux inhibitor, which can damage cells in the long term, but it's fine for the diagnostic process here. Outside of the U.S., it is not cleared for clinical use, but in the United States, it can be used for that purpose. It is available outside the U.S. through stem cell technology. All right. So those animal models suggest, and the clinical research suggests, that ALDH bright cells are important in humans and could be used in therapy of tissue damage and tissue regeneration. And in fact, ALDH bright cells have been used in 3 types of clinical trials. This has been done with clinical grade reagents. They've been safely administered to people in a cord blood trial and in 2 bone marrow trials. The cord blood trial was allogeneic cord blood transplant, unrelated cord blood in children. I think this is a very important, the reference to an abstract about this trial has been published, and you have the reference there. In this trial, part of a cord was sorted to get an ALDH bright population, and that was used as a supplement to the other part of the cord. It was a safety trial, and it's important in that these subjects in this trial were injected intravenously with these cells. The subjects were totally ablated in terms of their immune system, and no adverse effects were noted in the administration of these cells. Second trial there is an autologous trial, an autologous bone marrow used for critical limit ischemia patients. They had no option for revascularization therapy. A sample of their bone marrow was sorted to get ALDH bright cells. The cells were injected intramuscularly in the patient's leg that was at risk for amputation. The results of this trial have been written up now and will be published soon. They're in Press and Catheterization Cardiovascular Interventions. Again, no safety issues were noted in these critically ill patients. And the third one was another autologous ALDH bright bone marrow trial. In this case, they were critically ill ischemic heart failure patients with no options, and these cells were from bone marrow made the same way, and they were injected through a NOGA catheter directly into the heart muscle in the living tissue around ischemic area of the heart. And again, no safety issues were raised. Aldehyde has now announced that they have clearance for an IND for a clinical trial testing safety and efficacy of the same kinds of cell populations, autologous cell populations from bone marrow, in treatment of ischemic stroke. And in this context, the cells will be administered through the carotid artery. Now, again, these are the same diseases now that animal models suggested that these cells were efficacious in. Now these findings have been translated into the clinic by the Aldehyde group. These clinical trials now have demonstrated that donor cord and fresh donor marrow can be shipped to a manufacturing center for enrichment of ALDH bright cells. They can be manufactured under GMP conditions. The clinical doses of these cells can be manufactured and released within a day of receipt of the incoming material. And that these cells can be maintained and then be shipped back to the patients. The activity can be maintained during shipping for at least 3 days. As I've already said, the cells can be safely administered intravenously to ablated patients with cord, intramuscularly in critically ill patients with CLI, or through a catheter in the heart muscle for ischemic heart failure with no apparent safety concerns. So in terms of the feasibility and safety, there's good evidence now that these cells can be used. And in these trials, there have been signals noted that these trials weren't powered for efficacy, but there have been signals noted that are consistent with the improvement of perfusion in the ischemic diseases, and Aldehyde is intending to bring these products forward to treat these diseases in efficacy trials. So, to summarize what we've said, ALDEFLUOR can be used prospectively to isolate functional ALDH bright cell populations for research purposes. ALDH bright cell populations from different normal tissues contain different mixtures of stem and progenitor cells, and they seem to be appropriate for the tissue that they're isolated from. These cell populations are active in animal models of tissue repair. They express paracrine factors that may also contribute to tissue repair, so there are 2 mechanisms here. There's the classical stem cell model where the cells proliferate, differentiate, and form new tissue, but there's also a model where there's paracrine factors that can mobilize endogenous kinds of tissue activities, and both of these may be important in the clinic. The number of ALDH bright cells in clinical samples, clinical products, may give significant information and may be used and ALDECOUNT is clear to enumerate ALDH bright cells in the clinic in certain contexts. The trials show that ALDH bright cells can be manufactured under GMP conditions. They've been used safely to treat patients in several clinical trials. And additional therapeutic indications for ALDH bright cells in the clinic are likely to emerge from research studies that are carried on with ALDEFLUOR. ALDEFLUOR results can probably be translated efficiently into the clinic based on what we've seen so far. I have to apologize that we haven't had the time or the space in this format to cite all the people's work from many different countries who have used ALDEFLUOR and who've sort of created the body of information that I've put forward today. These are references where you can find these are just review references really where you can find more detailed information, and Stem Cell Technology does keep a comprehensive bibliography of work with ALDEFLUOR, and you can contact them for more information. I've enjoyed doing this. I'll be glad to answer questions.